you know about DNA. So I heard I already heard this. I heard Yep. I heard that. I heard deoxyribonucleic acid. I heard genetic material. I heard chromosomes. Julia, you had something else. There we go. I heard it's a double helix.
Nucleic acid.
So it's a nucleic acid. A nucleic acid was one of our 4 major classes of biomolecules from chapter 2. We had carbohydrates, lipids, proteins, and nucleic acids. So it's one of our 4 types of biomolecules.
Isn't this in just like lipids?
Not
RNA?
We have both DNA and RNA, but DNA is the major genetic component of all cells. It's a major genetic component of all cells. What's it made out of?
Isn't it like A, C, G, and T?
It makes protein, so it codes for protein. It's the recipe for proteins, but it's made out of A, C, T, and G.
I just took a vocab.
Hey, look at this. She doesn't have
I
to
have
do
a free
it
period.
Wednesday night.
You guys okay back there? Okay, just checking.
We were just getting things.
What else do you know about DNA? Have you heard it before?
Everything.
Most things.
Every living thing contains DNA. Even trees.
Decomposing things. Don't decompose things though?
Even little what's in there? Like just
Anything not living doesn't have DNA.
All cells.
So all cells having DNA means it's alive. Some things that are not alive have DNA. Viruses. Viruses are not alive. Viruses are not alive, but yet they have DNA. So that's a little strange. Not a computer virus. I mean like the flu virus. I mean like HIV, AIDS, that virus. Not a living thing, but has DNA. So there are some exceptions to the all living things. It's not so much an exception to the all living things have DNA thing, but some non-living things have DNA. Formerly living things, those have DNA, so dead things, decomposing things. You saw in the movie that we started watching last week a bit about DNA and something unique about it. How many of these A, T, C's, and G's do you contain? 6 billion. 3 billion pairs, so 6 billion total. Knowing that that's true, 3 billion pairs tells us a little bit about DNA structure. So before you guys get started on your activity for the day, I'm going to tell you a little bit about DNA structure, so hopefully it will help you understand some of what you read in your search. So You are absolutely correct in everything that we had on that previous slide. So if that's all your prior knowledge about DNA, 110% correct. Let's take a look at I don't know which picture is which without a thumbnail. Let's go with that one. Brilliant. Instead, we'll do it this way. So, if in your past you have encountered something like this when you talked about DNA,
So there's 6 billion of those.
most often you've probably been told that DNA is shaped like a double helix or a twisted ladder. The rungs of the ladder, so the steps on the ladder, are made of things that we call nitrogen bases. You're probably most familiar with those nitrogen bases by calling them A, T, C, and G. If you don't know what those are, do you remember from chapter 2, so like October, how many of you are like, I barely remember October as it is, let alone something specifically from a class in October?
I remember that.
Okay. So, do you remember the words polymer and monomer?
Oh, I remember power. I remember it says we yelled at.
So the polymer is DNA. Remember, poly means many. So all of the things that are strung together, it means it's many individual small pieces strung together, makes one big DNA. The 6 billion things that you have are 6 billion monomers. Those monomers are called nucleotides. Every nucleotide has a basic structure. Every nucleotide is composed of a phosphate group that we've labeled here as P, a 5-carbon sugar, which is deoxyribose. Hopefully from that chapter you remember anything that ends in OSE is a sugar. Glucose, fructose, sucrose. Deoxyribose, that means it's a sugar. And a nitrogen base. So we have a phosphate, a 5-carbon sugar, and a nitrogen base makes one nucleotide. 6 billion nucleotides makes you. So 6 billion of these individual things all strung together, actually 3 billion strung together, paired with its perfect mirror image, makes you. There are 4 choices to attach here at the nitrogen base. The choices are adenine, guanine, cytosine, and thymine. A, T, C, or G. You need those words spelled out? I'm assuming that's a yes. So last chapter when we watched that beginning of that movie, they talked about whole genome sequencing. And what whole genome sequencing means is taking one of your cells, splitting open its nucleus, and figuring out the exact sequence of A, T, C, and G that you have. And every one of you is different because your sequence of 3 billion A, T, C's, and G's is different. Now I just said your sequence of 3 billion is different. Your genome is 3 billion base pairs long, but it occurs as a double helix. So it has a mirror image, just like a ladder has 2 sides and 1 step. We pair together our nucleotides across from each other in 3 billion pairs to make all of your DNA, which is why you have 6 billion total. So they end up like this. Where there's an A here and a T there, we'll put a C there. It's going to pair with its complementary base directly across from it. C always pairs with G. So they're what we call complementary. They always fit together. C would have a matching G across from it. T would have a matching A, and A would have a matching T. And they fit together like puzzle pieces to make a perfect ladder. Then we take the ladder and we twist it, so it ends up shaped like that.
Can you straighten that out?
Your DNA?
Yeah.
This, I have no idea. I don't like touching it because it's like all dusty like. No, it appears that whatever student made this in like 1990 something.
1900.
But what you can't see. Remember the little boy in the movie who had a disease that they had to do whole genome sequencing? He was the one who got like was it the Green Lantern toy from his doctor?
They called him bubbly, was their adjective to describe him.
He looked like a effusive, bubbly young child. He was kind of cute, right? He seemed fairly annoyed that the doctor was taking so long to put together his toy. Of his 6 billion letters, so his 3 billion base pairs, one of them changed. One A or one T changed to a different letter. There was a single mutation which caused his whole immune disorder. So in this one letter, so the other 200,999,999,999 were all the same as you. One letter change caused an entire immune disorder that made him incredibly sick. So this spelling of the order of things in your DNA makes a huge difference for your health, for your traits, and for who you are. DNA is something that you encounter a lot in the news, and I teach human genetics, so this is kind of my personal pet peeve. Most of what you read in the news about DNA, people have no idea what they're talking about. Whether it's they weren't paying attention in their biology class, whether it's simply they just forgot what they learned because it was 5, 10, 15, 20 years ago. Most of the time you'll read it, and most, it doesn't really mean a lot. My challenge for you today for the rest of the hour is to find a current article from a news source involving DNA. And there's all kinds of stuff out there, and we're going to give you a list of things that you can choose from. This baffled me earlier. I'm going to give you a topic in DNA that you can research. I'll see if you know what it is. How many of you have heard of a GMO before? Oh, okay, see, I knew there were people. A genetically modified organism. So we can change their genes to read what it is we want them to say. An example of it, so those of you who've never heard of it can kind of understand what we're talking about. Tomatoes. Some of you guys probably like tomatoes, right?
No.
I hate them, so this example does not apply to me. Tomatoes. Seasonal thing in Michigan, right? Like it gets too cold, we can't grow them outside. There are places in the southern United States where they could grow tomatoes all year round, but they do get an occasional frost. Frost is enough to kill the traditional tomato plant. So what scientists did was injected a gene from a fish that lives in the Arctic Ocean. Injected it into the seeds. It essentially produces what is like an antifreeze. It's what prevents the fish in the ocean from getting too cold and freezing to death. Now the tomato expresses that gene and it's not susceptible to frost. We can do that. It's possible. They're doing the same thing with mosquitoes right now. Mosquitoes, you know what malaria is?
Yeah.
Mosquitoes are a vector for malaria. They can modify mosquitoes to make it so they are unable to be a vector for malaria. They can't pass the disease on. All they have to do is release the mutant mosquitoes into the environment, and then you have mosquitoes that can't carry malaria.
Just. She was gone. She's just gone.
And they can't transmit malaria. It won't work. So, what do you think? Those two instances, are those good reasons to modify something's DNA?
They're beneficial.
They're helpful to us, right? There are a few other instances in which we can do this. You will